Okay, so we will now be discussing first about the culture media, the different types of culture media. Before we start our discussion with culture media, let us define first some of the terms. Okay, now when we say culture media, we are referring to any substance that is capable of producing of any substance that possess the nutritional and environmental requirements needed for the bacterial growth. So culture medium is, or culture media would be the, culture media, it is the plural form, while culture medium is the singular form. Now, which means that this artificial media, or it could be a natural medium, would provide nutritional and environmental requirements. So this would include not only the nutrients or food, the pH requirements, the ideal or suitable temperature. So it's like the home of these microorganisms. Now, before we discuss further, let us define first the term culture. Well, bacteria are actually few organisms that have culture. Literally a culture. Okay? So, culture refers to group of microorganisms, and this culture is actually obtained from culture media. And this culture are divided into several types. Okay? So, we have, for example, if it's made up of one species, we call it pure culture. So, pure culture means there's no contaminant. Okay, it's pure culture. Mixed culture means that it is made up of two or more species. While when you say stock culture, this is the source of supply for industry, research, and for students' use. So, sometimes a stock culture can be classified further as ATCC strain, which you have to buy. Okay, American type culture collection. Okay? So, by the way, pure culture is sometimes known as the arsenic culture. So, if you hear the term arsenic, okay, it is similar as pure culture. Okay. Now, this is how we classify culture media. We could classify them according to consistency or physical state, according to composition, according on how the medium is dispensed, and according to use. So, let us, of course, you are familiar with agar, right? So, an agar is the same base that we're using for making a gelatin. Okay. So, an agar is a polysaccharide extract of the seaweed. Okay? So, galing siya sa seaweed. And the purpose of the agar is to solidify or to harden the culture medium. How do you prepare gelatin at home? You heat it, right? And then, if it passed the melting point, let's say for example, 45 degrees the melting point, beyond 45 degrees, it will be liquefied. And then, if the temperature drops, then it will be solidified. This is the basic principle when you are preparing gelatin as a dessert. Are you getting my point? Okay. Because we are using the same base medium. And that would be your agar. Now, according to physical state, we can classify culture medium according to liquid. Meaning, when you say liquid, it doesn't have any agar. So, okay. So, when you say liquid medium, it doesn't have any agar. So, what's the other name for liquid medium? Sometimes we call it broth. Okay? So, what are the examples of broth? Okay. For example, you're using BHI, familiar? Brain heart infusion. We're using nutrient broth, tryptic soy broth, thioglycolate medium. These are all examples of broth. Okay? So, Then, we also have semi-solid. When you say semi-solid medium, the semi-solid medium would have about 0.5 to 1.5% agar. So that's the composition of the semi-solid medium, about 0.5 to 1.5% agar. And why is it called semi-solid? We're usually using semi-solid if we want to observe the motility of the organisms. So if it's a solid medium, it's quite difficult to allow the organisms to move. So that's the reason why we have to use the semi-solid medium. So a semi-solid medium would have about 0.5 to 1.5% agar. So an example of semi-solid medium would be the MIL. This stands for motility indole lysine. And then another example would be the SIM. SIM stands for your sulfide indole motility. And then of course, majority of the culture medium that we're using, the plated medium, majority of them, we call them solid medium, which containing 2 to 3% agar. Okay, so these are the classification of culture media according to physical state. We can also consider them or classify them according to composition. So according to composition, we classify them as synthetic. If you know the exact composition of the medium, so you call that synthetic. Like those that are available commercially. Have you ever prepared a culture medium? Yes, right? So you have to look at the label. By looking at the label, you'll know the exact composition. How many percent would be the protein? How many percent would be the glucose, the carbohydrates, the pH indicator? Okay, so these are the important components because each of the culture medium would have certain function. Let's say, for example, which are the inhibitory substances, which would provide nutrients. Mostly protein would provide us the nutrients. Okay, what are the carbohydrate sources so that we'll know what sugar could a certain bacteria ferment. So that's the basic principle. Okay, non-synthetic means that you do not know the exact composition. By the way, these two are examples of artificial culture media. Okay, so these are examples of artificial culture media. Unlike this, it is a tissue culture medium. When you say tissue culture medium, it is made up of living cells. Okay, why do we need to have living cells? We need to have living cells so that we'll be able to allow the growth of obligate intracellular parasite. Okay? So that we'll be able to allow the growth of obligate intracellular parasites. What are these? Obligate intracellular parasites are those organisms that would require living cells for them to grow. Most common example are viruses. Okay, so viruses, you have to grow them in chick embryo. Cancer cells, such as the HeLa cells, this came from Henrietta Lacks. So, a woman died from cancer of the cervix. So, until now, her cells are still alive and are being used in different laboratories worldwide. So, it's actually a very controversial cell line because, first of all, you didn't get the consent of the family member. And the family members, I think, are not receiving royalties for using the cells of Henrietta Lacks. So these are examples of live tissue culture. But there are also bacteria that would require live tissue culture. For example, chlamydia. If you're still familiar with chlamydia, chlamydia would usually be cultivated using the McCoy cells. Okay, using the McCoy cells. Okay. Now, according on how the medium is dispensed, culture medium could be classified as plated or tube medium. Of course, plated means you place it in the dish that was invented by Richard Petrie. Petri dish. Petri. Actually, in some book, if you're watching YouTube from other countries, they're pronouncing it as Petri. But doesn't matter. But for as long as you know what we are pertaining to, so Petri dish. Okay. So tube medium, you place it in the test tube. There's an issue on preparation. Remember the rule. If it's plated medium, distribute first in Petri dish before sterilize first before dispensing. If it's a tube medium, distribute first in test tube. So dispense first before sterilizing. Obviously, if you're preparing a plated medium, we usually sterilize it first in Erlenmeyer flask, right? We autoclave it, and after autoclaving it, that's the time that we place it in Petri dish. Unlike if it's a tube medium, we first prepare them in test tube, and then after preparing them in test tube, then that's the only time that we autoclave them. So these are the basic thing. But the tube medium is further classified into 3. So we have the bath. So this is an example of bath. You know what I'm pertaining to, right? The bath. And then this is a bath slant. And then this is a slant, purely slant. So remember, whenever you see a bath, you prick a bath. I mean, not the bath of your classmate, okay? The bath of the tube using an inoculating needle. Halfway. For the bath slant, you stab, not prick, stab. You stab the bath halfway. You make a line here and streak. But if it's a slant, just make a line and then streak. Okay. Now, according to use, it could be classified as simple, enrichment, enrich, differential, selective, special, specific, and biochemical. Okay. When you say simple, it supports only the growth or multiplication of microorganisms, routine cultivation, and maintenance of microorganisms such as nutrient agar, BHI. So these are considered as simple culture medium. Okay? Basic requirement, peptone, agar, sugar. So sometimes there are no pH indicator, no inhibitory substances, which means that all of these microorganisms will be able to grow in the simple culture medium. Enrichment is needed to enhance the propagation of certain growth of microorganisms, such as the selenite broth, that is the enrichment for salmonella, or alkaline peptone broth is for Vibrio cholerae. Here's the thing. Let's say for example, you have a stool. Okay, and you want to isolate. Is this how your stool look like? Okay. You have a stool, and you want to isolate. Your question is there a salmonella in your stool? What if there are salmonella in your stool, and there are few salmonella in the stool? Majority of the microorganisms found in your stool, E. coli. They're overgrown by E. coli will overgrow and will outnumber salmonella, which means that if you're going to directly plate this, what you'll be able to isolate will be the E. coli, but not salmonella. Okay? To avoid this problem, we usually place the stool first in enrichment broth, such as the selenite F. The selenite F would usually have components such as the bile salt, okay, which will prevent E. coli from growing. So what will multiply here would be salmonella. So if you place them in a Petri dish, containing McConkey agar, what you'll have here would be Salmonella. Enrichment, so temporary cultivation. So the same principle if you want to use Vibrio cholerae. So instead of using selenite F, you use alkaline peptone water. Because of the pH 8.6, it prevents other organisms from growing. Are you getting my point? Enrichment, you call that enrichment. Okay? For Mycobacterium tuberculosis, that is also possible. You are using Bactec, you are using MGIT as enrichment broth. Mycobacterium growth indicator tube or Bactec for Mycobacterium. So that's also possible. Okay. Enrich will contain nutritive supplement needed for the growth of fastigious organisms. Fastigious organisms are those that require additional supplements in the form of blood, meat, albumin, vitamins, glycerol, potato. So examples are blood agar plate, which contains about 5 to 10% sheep's blood. And what's the difference between blood agar plate and chocolate agar plate? Okay, so blood agar plate is unheated blood. Chocolate agar plate is heated blood. But in choosing the blood to be used as blood agar plate, we're using sheep's blood or horse blood. But as much as possible, we do not want to use human blood because sometimes people are taking antibiotics. And then these people would have antibiotics in the blood, and that will be inhibitory for the growth of the microorganisms. So as much as possible, we do not want to use human blood. Okay. Differential. What do you mean by differential? When you say differential, this will allow the growth of one group of microorganisms and inhibit the growth. Hindi pala yun. When you say differential, we can distinguish the characteristic of the organisms by using the differential medium. Let's say, for example, McConkey agar is an example of differential medium. Because by using McConkey agar, we can classify lactose fermenters from non-lactose fermenters. So if they produce pink colonies, then they are classified as lactose fermenters. If they produce colorless or white colonies, then they are classified as non-lactose fermenters. Okay, so this is how we classify them. So we classify them according to the color of the colonies. So since we're able to differentiate characteristics of the microorganisms, so this is an example of differential medium. Okay. Selective. What do you mean by selective? Selective will allow the growth of one organism while inhibiting the growth of another. So that's why it becomes selective. So you add certain substances that will inhibit the growth of other microorganisms. Let's say, for example, you want to inhibit the growth of gram-positive organism. You do not want gram-positive organism to grow. Then you have to add gentian violet, sodium hydroxycolate, or bile salts. Or another example, you want to inhibit the growth of gram-negative organism. You do not want gram-negative organism to grow. Then you have to add potassium tellurite or sodium acid. Or another example, are you familiar with this? Yes, this is swarming of Proteus. You do not want Proteus to swarm. What are you going to do? Then you have to add alcohol or chloral hydrate. Okay, so the medium becomes selective. So McConkey agar is selective. So how do we classify McConkey agar? McConkey agar is selective because it can inhibit the growth of gram-positive while allowing gram-negative to grow. And at the same time, McConkey is also differential because it allows us to classify whether the organisms are gram-positive or gram-negative. Salmonella shigella agar. This is both selective and differential. Allows the growth of gram-neg and differentiates whether lactose fermenters or non-lactose fermenters. May bonus pa, if there's blackening, then there's a hydrogen sulfide production. Then, TCBS, thiosulfate citrate bile salt sucrose. Remember this one? This is for Vibrio cholerae. It's selective and differential. Because of the presence of pH 8.6 and the presence of bile salt, it allows the growth of gram-neg, but not gram-positive. Not all gram-neg will grow because of the pH 8.6. It prevents those that are non-alkalophiles from growing in TCBS. And at the same time, it is also differential because it allows us to classify whether the bacteria are sucrose fermenter by having a yellow colonies. By having yellow colonies, they are classified as sucrose fermenters. If they have blue-green colonies, then they are classified as non-sucrose fermenters. Are you getting my point? Okay. Special or specific. Sometimes, this one is also different, inhibitory. Let's say, for example, Lowenstein-Jensen for Mycobacterium tuberculosis is inhibitory because of the presence of malachite green. The malachite green prevents Mycobacterium from growing. Or, remember Muller-Hinton agar? Hindi Miller-Hinton. Meron ako nakita sa exams, Miller-Hinton yung sagot. It's Muller-Hinton. Okay? So, this one is special because for antibiotic susceptibility testing. Okay. So, this one, Lowenstein-Jensen special because for Mycobacterium tuberculosis. And at the same time, this one is also selective. Because of the presence of malachite green, it prevents other organisms from growing. Okay, so Muller-Hinton agar, this one is special. Then, another classification is biochemical. Biochemical determines the biochemical reaction of the microorganisms. Okay? So, this is how we, MRVP, metal red, Vodge Proskauer. So, I think you are all familiar with culture media preparation. Do you know how to compute for the ratio and proportion? Because the first procedure is to prepare all the necessary materials that are needed. And then, after that, you weigh the desired amount of agar using ratio and proportion. Kunwari, nakalagay dito, suspend 17 grams in 1,000 ml of distilled water. So, for example, the teacher asks you to prepare 200 ml only. What is the weight of the agar that you will prepare? 200 ml. So, you just do ratio and proportion. 17 is to 1,000. And then, the teacher asks you to prepare 200. So, all you have to do is to multiply 17 to 200 and then over 1,000. So, the answer is 3.4 grams. Am I correct? 3.4 grams. So, yun ang... So, 3.4 grams. So, you add 3.4 grams in 200 ml of water. And then, after that, you dissolve in distilled water and mix. Then you have to boil until the solution becomes free from undissolved particle. Do not autoclave if it's not purely dissolved, but it will burn and it will not look nice, and then it will not be solidified. Okay? So you have to boil it. But in our case, we have a microwave oven, so that shortens everything. Okay? But of course, you do not allow it to go beyond 1 minute kasi sasabog din yung, hindi na sasabog, but yung umaapaw sila sa Erlenmeyer flask. Di ba? If tube medium, dispense first before sterilizing. Alam niyo na yung rule na to, right? Plated medium, sterilize first before dispensing. So we sterilize in an autoclave. What's the principle of autoclave? Steam under pressure, 121 degrees for 15 minutes. Okay, so that ends our discussion on culture media preparation.